Thanks, Steve. It's really an honor for me to be here at the University of Washington. It's my first time in Seattle, and I have so many friends and colleagues here, so I just really want to thank everyone, and Steve especially for inviting me, and everyone for coming and welcoming here today. There we go. Okay, I always like to start with acknowledgements because I did not do any of this work by myself, and this work really comes from my interactions with patients. Actually, I trace this work back to my graduate work. I was a PhD student with Harold Varmus, and when we moved from San Francisco to the NIH, Harold would invite different scientific luminaries who were visiting, usually for NIH study section, into the lab, and that's where I first met Mary Claire King. And I think I was very imprinted by Mary Claire and her thinking, even when I was a young student. And I think that was percolating in my brain as I started seeing patients. And I talk a lot with my patients. I take their personal and family histories. I know their families. And I had this sense that germline predisposition was playing a role in my patients' malignancies. And this is a thank you note that one of my patient's daughters gave to me, thanking me for caring for her mother. It's a rendition of me doing a bone marrow biopsy on her mother, even though she was never in the room. I think it's kind of fun how, my arm just becomes the jam sheeting needle. But really, rather than them thanking me, I really thank the patients because the other half of my lab works on epigenetics and how cytosine modifications control differentiation potential of chromatin. But it was my interaction with patients and learning about their personal and family histories and starting to diagnose them that I feel like they showed me something that was not really appreciated in the field. At the time I was doing my fellowship, we had all of Mary Claire's work and her paradigm showing the importance of germline genetics in solid tumors and why that was supposedly any different from hematopoietic tissue and the bone marrow, which is under such a replicative stress, I don't know. But I really do thank my patients and their families. They've been very, very engaged in all this work, and they have led me on this path. And in the lab, there are a lot of people now working on the project. We are trying to understand basic pathways that are important, mostly in DNA repair in the bone marrow, and then also doing a lot of syndrome discovery, and I'll share some of that with you today. In the lab, Soma Das, and at the time Zexuan Li was at the University of Chicago, they were very willing to do something new, which at the time there really weren't comprehensive panels that would cover the diseases that we were interested in. You had MarrowSeq here. That was just being developed as a research tool first. It wasn't clinically implemented at that time. So Soma and Zexuan developed a clinical pipeline that we run now in the laboratory from human genetics. And so they were very, very receptive to this new idea that germline genetics might play a role in heme malignancies. And Jeremy is one of our pathologists who runs a pathology panel that I'll talk about that's more geared at prognostication and thought more to be a somatic panel, and we'll talk about that. And Dr. Vardaman has been very, very important in, I would say, getting the world community to recognize these syndromes. Dr. Vardaman and I looked over so many bone marrow biopsies from these patients, and we would argue about whether something was MDS or just every cell had this mutation and therefore the cells looked funny under the microscope. That he got very frustrated that his classification scheme did not accommodate for germline predisposition. And I got very frustrated with him one day, and I said, well, you're on the WHO committee, just change the classification. And so he took that seriously, and so he's the reason that there's now a chapter on germline predisposition to myeloid malignancies in the WHO. And we have collaborations with Anna, Hamish, Chris, Devendra, colleagues in Adelaide, other colleagues at NIH that have been very, very important. So I just want to acknowledge everyone's contribution there. So this case came to me on a Friday night a few months ago, and it's very represents a lot of the issues and very symbolic of the kind of cases that come to me. So I got home and, like neurotically check my email as soon as I get home. And here was this email from a doctor in Florida telling me that she's going to put the brother of her patient on an airplane to come to my Monday clinic 3 days later. And so I got rapidly embroiled in this story. So the patient was about 64 years old at the time she was diagnosed with acute myeloid leukemia. It had a normal karyotype. And molecular profiling had been done at a local hospital in Florida, and that identified 2 DDX41 mutations. One was a missense mutation at L373P with a variant allele frequency of about 50%. And then there was the R525H, which we recognize as a hotspot mutation that occurs somatically, and that was present at a variant allele frequency of 10%. This individual had been, or she is, a DES daughter, meaning that her mother had taken diethylstilbestrol when she was pregnant with her. And that raises this issue of environmental exposure and how patients' cancers, in my mind, arise both from germline predisposition as well as environmental exposure.
So did she have any of the other marks of DES exposure? The daughter, that is, like the vaginal scarring?
So apparently I'm supposed to repeat all the questions because the mics aren't on yet. So Mary Claire's asking if the patient had any other hallmarks of exposure to DES, and to my knowledge, she did not. This daughter is a lawyer. She's 29 years old, and she read the molecular pathology report, which was 10 pages long from this local hospital, and she PubMeded every gene and every variant. And she was pressuring the doctor, now at this referral hospital, that her mother had a germline DDX41 mutation because this VAF was about 50% and it was in the context of this hotspot mutation. And she was saying, there is no way you are using my uncle, who's HLA identical, until you tell me that he does not have the DDX41 mutation. And this is a phenomenon that I see again and again and again, that the patients and the families are very engaged and they are questioning the genetics of their family, and the doctors are either confused or they don't understand, or they've been taught that leukemia is somehow not genetic and they push back against it. But this doctor actually wasn't pushing back. She just decided, I'll put this brother on the airplane and Dr. Godley can figure it out. So now what? So the questions that are raised by this case, I feel like just it's a microcosm of everything we deal with. So how do we use molecular profiling results to find these families? And how do we test for germline status if we think that something is suspected? Because the easy tissue to get, blood, is affected by our diseases of heme malignancies, and I would say in all of us are affected by clonal hematopoiesis very frequently, and so blood, I would argue, is the last tissue we should be using, but we can discuss that. Now, if the brother has the mutation, and let's say we assume it's deleterious, which is a huge question, whether any missense, how can you determine the functionality of that? So if we assume the worst and say he has germline predisposition to a heme malignancy, and we give him GCSF to mobilize him to be a donor, what do we tell him about his risk for developing leukemia? What does GCSF exposure do? And what if he doesn't have the mutation, and we determine this variant to be germline in the patient? Well, what does it mean? Is it actually deleterious? Does it even matter? We don't have functional testing for DDX41, so what do? If the patient has a germline mutation and opts for allogeneic stem cell transplant using an unrelated donor, which I can tell you that this daughter, when I spoke to her on the phone, was what she was advocating for, she then wanted to know what I was going to do to confirm that the unrelated donor didn't have germline predisposition or that the cord didn't have germline predisposition. And she's right. Just because we don't see the unrelated donor, right, the cells come in a bag or the cord comes as a frozen unit, they have a genetic history. We just don't know it. And then what's the role of environmental exposure to cancer risk for people at an elevated risk? So this is the table that I use to summarize for myself all the currently recognized germline predisposition syndromes for heme malignancies, and my point is not for you to even be able to read all of them. It's to see how many there are. So there really are quite a lot, and every month or so I have to make the font size a little bit smaller on the table, because with exome sequencing and genome sequencing, we're identifying more and more of these syndromes. We are learning about biology all over the cell. I think we've known for many, many years that transcription factors play very important roles in hematopoiesis. We've known that telomere biology and DNA repair is very important. Ribosome biology is very important. The new paradigms that I think these syndromes have uncovered for us are found in these syndromes: ANKRD26, an ankyrin domain-containing protein; DDX41 that I've mentioned, which at least in Chicago is the most common germline predisposition syndrome that we see; SAMD9 and D9L. DDX41, for example, is found here in the nucleus as an RNA helicase. It's also found out in the cytoplasm, where it's part of innate immunity and sensing, along with CGAS and STING, and controlling interferon response. What function it has, meaning in the nucleus as its helicase function, or in the cytoplasm in the STING function, giving its, conferring the risk to heme malignancies, we don't know, but I'm sure that study of DDX41 is going to teach us new biology. I'm also pretty convinced that for every gene for which we see somatic mutations in heme malignancies, IDH1, IDH2, nucleophosmin, etc., there will be families who have deleterious inherited mutations in those genes. So it's just a matter of time. If we test the right tissues and analyze things the right way, we will find germline families for all of these genes, because the biology is reading out for you that they're important in hematopoiesis. So when I think of patients and I see patients in the clinic, I think of them at 4 different levels, if you will. Baseline genetics, the baseline epigenetic changes that they have, which often come with their ethnic background. The acquired genetics that are occurring in the stem cell or in the tumor. And then often, at least for leukemia, those acquired genetic changes are giving us acquired epigenetic changes in the tumor that are giving the phenotype. And I believe that only when we understand all of that in context together, we'll be able to develop effective treatment for patients. So how do we recognize patients with germline susceptibility to myeloid malignancies? And one question related to that is, how many people actually have germline predisposition? So, all comers, how many patients are there? So I'll try to answer that, at least for some patients, for you today. So what do we pay attention to in our program at the University of Chicago? We try to have a high index of suspicion for all patients that we see. And the way we do this mostly is at the time of the bone marrow biopsy, because the bone marrow biopsy is when we have access to the skin, we're cutting into the skin, and we have access to the bone marrow. So it becomes a unique moment where we can get the skin sample, which we use cultured skin fibroblasts as our standard for germline DNA. And we access the affected tissue at the same time. We try to be systematic. We try not to guess what gene this might be. So we do panels, and the lab is actually moving to whole exome shortly. Consider all of the data available, which for us increasingly and now very regularly includes molecular profiling. So we do a panel that's run out of our pathology department that's supposed to help prognosticate. Now that panel has about 150 genes on it. The actual panel that gets run has 1,200 genes on it, and we'll come back to that in a minute. But the 150 genes that get read out, at least I was involved in that 150 gene list, and I pushed all the genes that at least at that time were known to confer germline susceptibility into that 150 gene list. So that becomes very helpful, and I'll share that with you. Over time, as we run that pathology panel to define if someone has molecular mutations that will prognosticate for their leukemia or other heme malignancy, or define molecular remission, any variant that you see every single time, whether someone has disease or doesn't, is a surrogate for germline mutation. In fact, I would say for most of these syndromes, basically every time we see an invariant variant, we have then confirmed that it's germline, and I'll share that with you. It's very important that the testing be able to find copy number variants, because for our patients with heme malignancy, inherited gene deletion mutations are very common. And on the pathology panel side, where they're just doing next generation sequencing, they generally miss the copy number variants with the bioinformatics that they do. So when we're studying a patient for germline susceptibility and we're running our human genetics panel, I'll show you that that panel actually has 2 parts, and it's specifically designed to allow us to find the germline CNVs. Yes, we have to know the predisposition syndromes, but as I said, we actually don't try to guess, and we do more and more by panel, and soon we'll be doing everything by exomes. So what are the key features that we look for? First, multiple cancers within a single individual. So I think the most number of cancers I've seen in a patient referred to me was 5. On his 5th cancer, which was a leukemia, somebody thought maybe he has a germline predisposition. It was BRCA1. It was pretty incredible. 5. No, now it's 2 for us. If we do a bone marrow biopsy, we take the personal history and that person has had any other cancer, we do the skin biopsy and the cultured skin fibroblasts and germline testing. Now we've talked about therapy-related leukemia for a long time, right? A leukemia that develops in someone treated for a prior cancer or immunosuppressed for some other condition. But actually, we have more patients who have what we now term double cancers. That's where they have the leukemia first, and then they get the solid tumor. So I was talking the other day, we have a patient with systemic mast cell disease, and he came to clinic the other day with a stage 3 melanoma. So when we've run clinical testing on patients with therapy-related leukemia, about 20% of them test positive. If they have double cancers, it's 25%. It's a little bit higher. I don't know if it's statistically significant or not. But anybody with multiple cancers is reading out a cancer predisposition for you. If the person in front of us has a blood cancer, that's why they're having the bone marrow biopsy, and they have any other blood cancer or young onset, which we define to be pretty young, solid tumors, within 2 generations, we go ahead and do the clinical testing. If they have other heme abnormalities within the family, these are much harder for people to know about than we would do. That like cytopenias, that like platelet, thrombocytopenia, low platelets throughout the family, we would count that as well. For us, at least for heme malignancies, we do not look at age of onset. And I do think that this is somewhat of a bias in the way we've been taught. We've been taught that if someone presents very, very young, they are likely to have a germline predisposition, and that is true. But it doesn't mean that somebody who's presenting with a heme malignancy in a normal age group, like 60 or 70 years old, doesn't also have germline predisposition. So I think we've been focusing on the young people and ignoring people in what we consider an average age group of presentation and thinking that somehow that means they don't have germline predisposition. And the reason I say that is we're testing lots of 60, 70, and 80-year-olds and getting germline predisposition. And the phenomenon of anticipation we see very strongly in these families.
Would you care to comment on the percent of people that you test 60 to 70 or 80?
So, yeah, I'm being asked what's the percent of positivity across the age range, and I don't have the answer. We haven't done it systematically, but that's what I'm going to show you is one of the proposals I have to extend what we have done. So I'll get to that question in a minute. Involve the nurses and genetic counselors. They're incredibly smart people, incredibly well trained. Unfortunately, I think a lot of them are not trained in germline genetics of heme malignancies, so we train people locally and they're very, very helpful. especially pre and post testing and in preparation for HLA matched related stem cell transplants. So we're really trying to tighten our pre-transplant workup for anybody being considered for a related donor. Take a very careful personal and family history and look at the molecular profiling that's been done on the patient. So this is the algorithm that we have for patient workup. I indicated to you, we interview every patient at the time of bone marrow biopsy that's inpatient and outpatient for personal and family history. And you would like to think that the clinic doctor has done this already, or the inpatient house staff has done this already. But it's not the same as somebody who's just thinking about that. So we have one employee who literally goes to every single bone marrow biopsy, and he takes that history and he knows when to do a skin biopsy. He and I review the outpatient schedule every week, and we look at the molecular profiling, which I'll share with you, and we kind of predict who's going to need a skin biopsy. But then he goes and does that on all the patients. And it's incredibly helpful. He told me the other day that on an average day he walks 10 miles, which made me feel really guilty, but he's running all over the place getting this work done. Exactly. So I told you that we do routine clinical testing of leukemias, and we do a lot of bone marrow biopsies on myeloma. That molecular test is often done on those patients as well. Anytime we see a variant in any gene that could be germline, we will go and get cultured skin fibroblasts or appropriate germline tissue and test. And then, as I said, we're really trying to identify people at the time of matched related donor transplant prior to the transplant. It's a very difficult conversation to have with someone, as I have had to have, after they've been a donor, and you tell them they have a germline predisposition to a myeloid malignancy and you've given them a myeloid growth factor. That is not an easy conversation to have. So we would like to not, have that after the fact. So we'd like to know this up front. So as I indicated, when we do a bone marrow biopsy, we cut into the skin already. That's our standard practice. So it's quite easy to do the skin biopsy right at the time of the bone marrow biopsies. Patients don't care that their scar is a little bit bigger. And so it's very easy to do. The skin goes to the clinical laboratory. They grow the cultured skin fibroblasts. Most of our patients consent to our research, and so we get a flask of the skin into the research lab later, and that becomes an incredible reagent to make iPS cells and all kinds of other different assays you can do with skin, so it's very helpful. We isolate genomic DNA from cultured skin fibroblasts, and then we run this pipeline that has two parts. One is a next generation sequencing panel that covers all the genes known to confer cancer risk, and the other is a microarray. It's a custom designed microarray with very densely tiled oligos that cover these genes, and we find lots of gene deletions, either partial deletions like single exon deletions or deletions that cover several exons, or sometimes even whole gene deletions. So if people test positive, then we do very dense family-based genetic counseling, cascade testing, and site-specific testing, as you might imagine. If they're negative, they go into our research. And really, if we had all the money that we would want to have, we would just sequence everybody, because I'm sure that a lot of syndromes exist that we have not yet described. Now, what's the yield when we do this with the criteria that I shared for you? For the first about 200 patients that we tested, you can see we are looking across the entire age range of life, and the answer is about 20%. Really doesn't differ that much between children and adults. So I would say 1 out of 5 people is pretty common. Now, what do we do with these variants that we find on clinical testing? So first, I really want to thank David for being the co-chair of this ClinGen committee that we've been working on for a couple years now. He's been a wonderful, wonderful partner in this group, and it's trying to help us interpret variants that we see. We have 2 committees that are co-sponsored by ASH and ClinGen, one on inherited thrombocytopenia, and David and I co-chair the committee on inherited myeloid malignancies. We started with RUNX1, And it took us about 1.5 years to do curation rules for RUNX1. We were challenged there by knowing that there were a lot of inherited deletion mutations in RUNX1, and yet there were no general curation rules for germline deletions. So we came up with our rules, and then the parent ClinGen committee came out with recommendations that were in line with ours. And now we've moved on to GATA2, so we're working on developing curation rules for GATA2. And the challenge here is that we already know about an enhancer region within the GATA2 gene where you can have germline mutations that affect the expression of GATA2. And yet there are no general rules in ClinGen for annotating variants within enhancers. So we are working on that. So our ClinGen committee has been really exciting. We've branched into new territory, and now we have scientific ideas of working on GATA2 syndrome. So it's really been a tremendous committee to be involved in. I really want to thank David again for all of his leadership. You can see the results of our work for RUNX1 in ClinVar, so we're annotating all of the variants that are listed in ClinVar using our rules. Now, I talked about that we use this pathology panel every time we do a bone marrow biopsy on a leukemia patient, and it defines for us molecular remission. But obviously, the germline is not going to change, right? The germline is always there. And so we wondered, when you see a RUNX1 mutation, how often is it germline? So we took, for the first 1.5 years, we were running this pathology panel, any variant called in any gene that conferred risk for heme malignancy, we went back and got true germline tissue from those people. And for our panel, the only time we saw a germline mutation was when the variant allele frequency was over 40%. But recently, one of my DDX41 mutation carriers, and I know she has germline DDX41, just had a bone marrow biopsy, and her variant allele frequency on that run for DDX41 came out at 38%. So we prioritize patients who have variant allele frequencies over 30%, because I don't think that the test is always perfect every time. We found 4 variants over 40%. Germline were present 24% of that time, so it's common. And the new way that we're analyzing this, this is, for me, a little bit of a frustration because I think the pathologists who are writing the reports and tell us the variant allele frequency should also be telling us if that variant has been seen every single time that patient has had that study. So if they see a P53 variant 10 out of 10 times, I want to know it's 10 out of 10, not 2 out of 10. But so far, my pathology colleagues say that's my job to do. That's the clinician's job to do. The problem is that the reports come out statically. So in the computer, everything comes in chronologically. So in order to do that, I have to alphabetize all the tests under pathology, scroll down to this test, and open each test individually. Most of my colleagues are just not going to do that. But in the lab, we are trying to find these patients, trying to use these data and not miss the patients. So we are graphing the VAFs over time. And for a gene like P53, you see that most of these lines are kind of all over the place because in heme malignancies, most of the time P53 mutations are somatic. So as the people have disease or don't have disease, the VAF goes up and down. But sometimes the VAF is right around 50%, and those people have Li-Fraumeni syndrome. And all of the patients we've identified with Li-Fraumeni syndrome would never have satisfied any clinical criteria for Li-Fraumeni syndrome. I don't know if you agree with this, Mary Claire, but I think from my perspective, these clinical syndromes were described by clinicians in the clinic, and they were naturally going to find the strongest phenotypes. Yes? And now we...
That's true for every genetic disease.
Exactly. It's true for every single genetic disease.
They're all described at the worst because that's what goes to the doctor.
Exactly right. So I'm glad you all agree with that. So that means when you look at it from a molecular standpoint, you see stuff that doesn't fit. But I think it's all relevant. So now we graph these, and you can just read out the germline. BRCA2 is not typically somatically mutated in leukemia. So sometimes you get people who go up and down, but mostly you have germline. So we have used this analysis for the 150 genes that get read out clinically and actually defined a new syndrome, found a new syndrome this way. So CSF3R, yeah.
So you kind of showed a graph of your leukemics that come in.
These aren't new, so they're just every time. It could be somebody who comes to us with relapsed disease, but once we have them in our system, if we do 3 bone marrows, they will have 3 molecular tests.
Yeah, so my question was, the individuals that come to your clinic, is it only those individuals that meet those criteria that get into your clinic that get germline testing, or are you able to do this programmatically? And how does that, is that part of the automatic testing that happens to the person who gets a bone marrow aspirate and biopsy, just like we here would automatically get somatic testing?
Exactly. So Shoban's saying, why don't you do germline just like you automatically do your somatic testing? And we just haven't implemented that yet. That is, to me, the obvious next step. So if our criteria are the individual has 2 or more cancers, or they have 1 other heme malignancy within 2 generations, or young onset solid tumor, that's still a lot of our patients. So we do a lot of skin biopsies. We probably do 5 a week. We probably do 50 bone marrows a week. So it's not everybody. The reason I haven't implemented it a little bit, maybe is depressing, is for insurance. So insurance companies generally don't recognize our diseases yet. We haven't had the advocacy of the BRCA community over the generations. And so the insurance companies don't have a policy. As soon as an insurance company doesn't have a stated policy, they consider anything to be experimental. And if it's experimental, they don't have to pay for it. So the human genetics lab that's running our germline testing has a very low reimbursement rate. Our hospital administration isn't so happy with that. And if I do testing on absolutely everything, that phenomenon will get worse. But I'm pushing the system, as you might imagine, and I hope soon that's exactly what happens. I have to have data to argue that it's worth it, and I'll share a little bit of that with you. But we haven't done it on everybody yet. But if you had all the money in the world, and some centers are doing that, you know, germline and somatic on every person walking in the door. So if we do this kind of analysis, we call this test OncoPlus. If we do the OncoPlus analysis over time for all 150 genes, we read out all the syndromes you would recognize as being cancer predisposing. JAK2 comes out in this mix because when we treat a patient for a JAK2 mutated myeloproliferative disorder, we don't try to get rid of the JAK2 mutation, or at least our therapies are not effective in getting rid of the JAK2 mutation. So they just always have the mutation. You'll see ASXL1 here as well. I don't know if that's like JAK2, it's just always present. All of these patients, I went back and looked, all of them had disease at the time of the bone marrow biopsy, so nobody was in remission. But we have germline tissue on some of these people, and we'll look. But you'll see that CSF3R is right here in among all of these other syndromes that you would recognize. And so we looked at CSF3R and we're able to do functional testing with Belinda Avalos, and 2 of these variants are germline and affect the CSF3R protein, make it abnormally in function. And so we're pretty convinced that CSF3R germline mutations are a new syndrome. It's pretty rare, but it's definitely there. Now what we want to do is go back to the pathology panel, which is 1,200 genes, and run this analysis on all 1,200 genes. So that's going to take a little bit of time for the bioinformaticians, but I estimate we'll find another 15 syndromes or something.
What type of mutations are they? Because as you know, where in the molecule?
I can show you that later, Marshall, yeah. So back in the day, maybe 4 or 5 years ago, when we were trying to answer the question, how often does somebody have a germline mutation? I tried to think of a patient population where you would actually see this pretty frequently, to try to tell people this is not incredibly rare. And so this is work that we've been doing with your center here with MarrowSeq. MDS is a disease of the elderly. Median age of diagnosis is in the 70s. So why should a young person, somebody down here, be getting MDS? Most obvious reason is they have a germline predisposition. So in order to test this, whenever we could, we used cultured skin fibroblast DNA for this assay. Sometimes we use cultured mesenchymal stem cells, and I've been challenged on this point that these people, most of these bone marrow aspirates were done at the time someone was diagnosed with MDS, and people have shown that the mesenchymal stromal cells, which are non-hematopoietic, but they can have mutations in them. And so I just want to, for those people who know that and are paying attention to that detail, we only would call a variant germline if we saw it in the cultured MSC and in the hematopoietic cells that we have from that aspirate. So 2 lineages would have to have that variant in order for us to call it germline. Very rarely we use saliva. For us, we really don't consider saliva to be germline because there's so many hematopoietic cells there. Paraffin-embedded tissue always has blood in it, and we can read out variants from donors after transplants. We can read lots of, say, clonal hematopoiesis associated mutations in paraffin-embedded tissue. So again, we used very few of these, and obviously T cells are hematopoietic, so we only did that on 1 patient. And essentially, from about 120 samples that we got from all over the world, because obviously young MDS is very rare, bottom line answer is about 15% of these people have mutations. And what are these? And let me say, when I say they have mutations, this 15%, this is very conservative calling. So these are variants, and they're here. So hopefully you agree that these are deleterious mutations. They're in telomere biology, and they're in DNA repair. If we said any variant in any gene, not something known benign, but let's call them variants of unknown significance, in any gene, in any pathway known to confer germline susceptibility, that percent positive would go to 80%. So the real answer is somewhere between 15% and 80%. Now, it's not going to be 80, but this is much more common than what we think. And we know that pediatricians have essentially done this work in pediatric MDS. So the answer, if you are from 0 to about 25 and present with MDS, the answer's got to choose SAMD9 and D9L. From our work, 18 to 40, we get BRCA and telomere biology disorder. And we know that older people get DDX41. And so in the end, the age of presentation is only a surrogate for the biological pathway. If you have a germline mutation in a pathway that gets to MDS quickly, you present as a child. If it's a little bit slower, you present as a young adult. And if it's slow, or if there's some other factor that we don't understand about DDX41, you present as an adult. This gets to Eli's question. What about the entire age range of life? Is the answer always about 15, 20%? That's basically what you get in the pediatric age group as well. Or is the percentage declining as the age increases, some steady decline? Or is there actually some sort of inflection point after which we really don't have to worry so much about these mutations? This is important for thinking about screening donors in a very systematic way. And so we would really like to repeat this study in every age group from 40 on up to figure out what the frequency looks like. So hopefully one day this will get funded and we'll be able to answer it. Now, how do you go from having a germline susceptibility to developing any cancer? And I know it's focused on myeloid here, but let me just pause to say that I don't think there's anything sacred about the myeloid lineage. all of germline predisposition applies to lymphoid malignancies as well. We just have more information right now about myeloid, but lymphoid, if anything, from what we've seen, is even more genetic. So the way I see cancer, I've already kind of talked with you about this. I think everyone has an element of germline predisposition and environmental insult. There are some germline predisposition mutations that are so penetrant that you don't need much environment. And for example, germline CEBP alpha mutations are highly, highly penetrant. At least the 5' end mutations are essentially 100% penetrant. So if you have that mutation, at some point you are going to get a myeloid malignancy. There are other malignancies that are very, very environmental. So we think of post-atom bomb radiation as conferring a huge risk to leukemia. And I used to think we were aging out of people who were exposed to radiation. But in Chicago, actually, we have a lot of Chernobyl survivors. And they get a lot of leukemias and other cancers. And actually, my patient who has cleidocranial dysplasia, germline RUNX2, her grandfather had leukemia. And I said to her, oh, not only do you have a de novo germline RUNX2 mutation, but you also might have another variant coming from your grandfather. She said, no, 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 my grandfather didn't have inherited leukemia. He worked at Los Alamos. And he was there in the 4 a.m. experiments where the night sky turned, bright white, and all 15 people who were there got leukemia. So he had atom bomb level exposure. And we still tested her for germline predisposition, but I think, unfortunately, there really are people who are either related to others who had atom bomb level exposure, or even those people today. I've talked to you about therapy-related myeloid neoplasm as being kind of a classic example, if you will, of someone potentially with germline predisposition who then gets environmental exposure. And this became first apparent to me when Jane Cherpek did her study at the University of Chicago on women with who survived breast cancer who went on to get a therapy-related leukemia. And Jane got germline DNA from those people and just did a panel that was very BRCA focused. And she could show that about 20% of people had a deleterious mutation in one of these genes in this pathway. And some of these people who tested negative, we now know actually had disc keratosis congenita. So they had germline TERT mutations. So again, it's at least 20% and maybe more than that. And I hope, and I'm sure here, I'm preaching to the choir a little bit, but this idea that we have that there are GI syndromes or, the breast cancers, cancer risk, it's not about the tissue, it's about the biological pathways. So I hope in the future our cancer risk groups or clinics can be focused around biological pathway because these pathways are affecting these different tissues in different ways. So at the time when Jane found this, we thought, well, that's really interesting about BRCA1 because only BRCA2 at that time was known to be a Fanconi anemia gene. And so Jane worked to knock out BRCA1 in the bone marrow. And those mice get spontaneous leukemias and lymphomas. And the reason I put it in air quotes, the spontaneous part, is what we were doing was putting those mice on the shelf and bleeding them every month. And we don't think of bleeding a mouse as being a large stress hematopoiesis on the mouse, but it is actually. So since we've published this work, we went on to knock out BRCA1 and just leave the mice on the shelf and not bleed them. If we don't bleed them, they don't get tumors. We can time the tumors by when we start bleeding them. So if we start bleeding them at 6 months, within 4 or 5 months, they all get leukemias and lymphomas. So there's something about driving the hematopoietic stem cell into division. We think it has to do with backup DNA repair pathways that are error-prone, introducing mutations, chromosomal rearrangements. These tumors are very, very chromosomally abnormal, which for a mouse is unusual. So right now, there's a graduate student, Stephen, who's working very, very hard on understanding DNA repair pathways in bone marrow. And this is where I think we can have some really important tissue-specific differences and why it's important to understand these genes and proteins in the tissue context for each tumor type. Now, in a person with germline mutation, all the cells of the body have that mutation. I don't think anyone doubts that a RUNX1, GATA2, CEBP alpha mutation and master transcription factors for hematopoiesis don't play a role in the hematopoietic stem cell. Yes, they have to. But what about all the other cells in the marrow? So the bone marrow is a very complex organ, as you know, and the hematopoietic stem cell is one cell being supported by all of these niche cells. And so we've been using the mesenchymal stromal cells that we first get from the patient's bone marrow and asking those cells to differentiate down different pathways. And they're very, very easy to tickle with different additives to the media and get those MSCs to differentiate down particular pathways. And I don't really want to go through all the details because essentially each one of these bars represents MSCs from a patient with a different mutation, and they have their own unique abnormality. So the MSCs, because we're asking those cells to differentiate and require transcription factor function, they often read out as being abnormal. Now, that doesn't prove that that is contributing to the disease, but I think it's very suggestive that the germline mutation may have importance in cells outside just the hematopoietic stem cell. Now, I just want to take a few minutes to talk about acquired genetics. And we're very interested in how someone with a germline mutation goes on to acquire secondary mutations and get leukemia, and also how that happens in people without germline mutations. So one study that we've done a few years ago was looking at the incidence of clonal hematopoiesis in our germline mutation carriers. And essentially, we can see clonal hematopoiesis very, very easily in patients with most of these germline mutations. And so we imagine that they are acquiring mutations throughout their lifetimes that's giving them a risk for leukemia. The one syndrome where so far we do not see clonal hematopoiesis, at least for the genes that are classically defined as clonal hematopoiesis, known oncogenic drivers, is DDX41. Now, DDX41 is very, very different. I told you that the age of onset is very different. It has function in the nucleus and the cytoplasm. For us, it's the most common germline mutation. And in asymptomatic carriers who are just living their lives well before they have any malignancy, we have not seen any clonal hematopoiesis in these people, which makes me think that there's something else that's happening in their lifetime that's giving them the stimulus. It's very, very interesting. We don't have the time to go through it, but this is one of the syndromes where we see founder mutations. So the A500 frame shift is seen in Asian populations, especially in Japanese and Korean patients. The D140 frame shift and the M1I are seen in Caucasian patients. And regardless of which germline mutation is present in the leukemia, you often see acquisition, especially of that R525H. So I would argue for that first patient I showed you on that first slide where she had a missense mutation in DDX41, but R525H at 10%, the fact that she had a hotspot acquired mutation, to me, argues that it is a deleterious DDX41 mutation. It's circumstantial evidence, but to me, that matters. Now, DDX41 is interesting in so many ways. We think that all you have to do to cure a patient with a germline predisposition is replace the hematopoietic stem cell. Do an allogeneic stem cell transplant from a non-mutated donor. For germline RUNX1, GATA2, CEBP alpha, you can essentially correct their hematopoiesis by introducing wild-type stem cells. However, for DDX41, and this is anecdotal, but we have several patients where we know they have germline DDX41. We've told them to have either an unrelated donor or a related donor. We know they're DDX41 wild type. They are not doing well after transplant. That suggests that their DDX41 mutated microenvironment is not able to provide normal signals to the wild-type stem cell. And I'll just show you one example. I'm also not sure what's going on with DDX41 in terms of the size of the families. We talked about this earlier. There are very, very large families with DDX41, at least the families that I have. So this is a family with 15 siblings. It's a fascinating Japanese-American family. And this individual lives in California. We diagnosed him. We said, don't worry. We know you have DDX41. You can have an allotransplant. Everything's going to be great. And it wasn't so great. He had an unrelated donor transplant, and after transplant had a neutrophil count of 200 and a platelet count less than 10,000 for 1 year. More than a year. He was getting 3 times a week platelet transfusions. So we traded one horrible disease for an absolutely horrible quality of life. And during this year, I was feeling incredibly guilty because I had not made him better and was really thinking a lot about inflammation, the role of inflammation in the bone marrow. And I started thinking that maybe we should try eltrombopag or Romneyplasti, that maybe this microenvironment was producing inflammatory cytokines that were suppressing the normal wild-type stem cells. So I begged his doctors to try Romneyplasti. I said, you cannot make him worse. His platelet count is 5,000. And finally, after more than a year, they tried. And it was pretty remarkable. Within 1 week, he had a normal neutrophil count and a normal platelet count. He went on to get Romneyplasti every 3 weeks for several months, and now he's weaned off it. He's doing extremely well. We have about 3 other cases where this exact same thing has happened. That patient that I showed you in the first slide, her brother was DDX41 wild type, but I told them, you're not out of the woods. You're going to transplant with his cells, and about day 60, her platelets are going to crash. Just give her Romneyplasti. Don't argue, just do it. And the daughter made sure that happened, and as soon as the Romneyplasti was started, she's done very, very well. So it's not proving the phenomenon, but I hold out the fact that these germline mutations in cells other than the hematopoietic stem cell could have an effect. At least the signal we see is in DDX41. I only have a couple more minutes, so I'm going to try to go really fast. We're really interested in the process of clonal hematopoiesis. And we use the TET2 deficient mice as a model for that. It's not a perfect model because people are somatic mosaics for these mutations, and the mice have a VAF of 100%. But what Banajabri at University of Chicago was able to show was that the mice that are TET2 deficient, the mice that go on to get what looks like a chronic leukemia, it's like a chronic myelomonocytic leukemia, are the ones that have translocation of microbiota from the gut into the systemic circulation, causing an IL-6 dependent inflammation. And those are the mice with the high IL-6 levels that go on to get the chronic leukemia. If you make the mice germ-free, or you treat them with antibiotics, or you treat them with anti-IL-6 antibody, they will not get leukemia. Now, we don't know if that's happening in people, but we're quite interested that, again, another, if you will, environmental exposure to our microbiota could be driving leukemia, at least for some of these patients. I'm just going to run through this really fast to get to this part. So the way we've been trying to identify clonal hematopoiesis is the only way I can think of doing it, because remember, if you have clonal hematopoiesis, you have normal blood counts by definition, so you'll never just find these people. So we get people at the time of hip replacement. These people have osteoarthritis. Again, air quotes, that's at least what's in their chart. And we get their preoperative blood, which is collected about 3 weeks before their hip replacement. Then we get this sample, the hip itself. It comes into the lab, and we collect mesenchymal stromal cells, hematopoietic cells, etc. And it's been really interesting. First, I had no idea that people in their 40s and 50s had so many hip replacements. But even with this small number of patients, we can already see a statistical significance with prior chemotherapy. We have been defining CHIP using the Cornell Precise 3 panel because it has very high depth. Well, I think 2000x is high. You may think that's low, so it's all relative. But it's about 2000x coverage over every exon in every gene that has been defined for clonal hematopoiesis, plus some other genes that are important in atherosclerotic disease. And out of the first 35 patients, 14 of them had clonal hematopoiesis, so 40%. And the average age was 68. So this is about twice what you would expect. Now, maybe that's an artifact of the assay being comprehensive and deep, or maybe there's something about the fact that these people have arthritis. What's the chicken, what's the egg? Is the clonal hematopoiesis causing a local inflammation that's getting read out as arthritis, and that's why they're getting hip replacement? Or is the arthritis a local inflammation that's allowing mutations to persist in the hematopoietic stem cell? I don't know what's the chicken and what's the egg, but we're testing the blood after hip replacement to see if when you physically remove that bone, do you remove the site of clonal hematopoiesis? Can you actually cure clonal hematopoiesis with hip replacement? And I was thinking I would have the answer for you today, but I don't quite have it yet. So I don't really know. But again, the patients have been incredibly helpful. We call the patients several months after their hip replacement. They remember having donated their hip to science, and they literally drive to our center and donate their blood for after the post-surgical specimen. So I just want to thank the patients again. Thank you for all of your attention, and happy to take a few questions.
So let's say you find somebody who's 65 and they have an MDS, or a germline RUNX1 mutation. They're one of the 15% or whatever that are like that. And you compare their disease to somebody who does not have a germline mutation, but the same RUNX1 mutation. Do they differ in platelet count or marrow blast? And after you account for those things, does it matter that it was germline in terms of their product?
Yeah, so Siobhan and I were talking about that first thing this morning. So it hasn't been looked at systematically. I think you've done your own local analysis here. It has been looked at, say, for CEBP alpha, germline CEBP alpha. Those patients are very chemosensitive, so they go into remission just like, we consider biallelic CEBP alpha mutations to be good prognosis. 10% of those patients have one of th' mutations, usually the 5' mutation as the inherited mutation. But they have relatively chemosensitive disease. Our experience is that DDX41 mutant patients also have very chemosensitive disease. So you might think that having a mutation somehow is a terrible thing in terms of chemosensitivity. But again, I'll turn to germline CEBP alpha. In those patients, they usually inherit the 5' mutation, and they acquire a mutation on what was a wild-type allele at the 3' end. So the 3' end mutation is a molecular marker of the leukemia. So Jude Fitzgibbon looked in relapses of patients who had germline CEBP alpha mutations, and that 3' end mutation was distinct, was different. So those are not relapses, they're second and third primary leukemias. If you look at the outcome of patients who have biallelic CEBP alpha mutations in which one of those mutations was germline, or 2 acquired CEBP alpha mutations, the germline patients do better. The only difference between those 2 people is not in the hematopoietic stem cell. The hematopoietic stem cell has 2 CEBP alpha mutations. It's the microenvironment. So when the microenvironment is CEBP alpha mutated, those people do better, suggesting they're more chemosensitive. So that's another piece of evidence to suggest that the germline mutation, which is present in all the cells, can confer properties to the leukemia. And things are either the same, or I would say, as a general statement, these people are more sensitive to chemo. But they're not worse from what I have seen, and I think your experience here is similar to that.
I want to ask you about whether we should anticipate the following unhappy scenario. As we're saving women who are, let's say, BRCA1 mutation carriers from developing breast or ovarian cancer, and so they never have cancer, they grow older, do you think that we will be seeing an upsurge of leukemias in these BRCA1 carriers?
You already, well, what's the black box warning for PARP inhibitors?
Exactly.
It's leukemia.
But of course,
Is that the PARP inhibitor? Is that the fact that they're...
But I'm talking about women who never get cancer, so they never have to use it, right? So they're well, and they will not get breast or ovarian cancer, but they will get older.
Yeah,
So do you think
yes.
these will come
Yes, I
here?
do.
Yeah,
Yeah.
I do too.
And I think you're already seeing it in the PARP inhibitor population.
But that's the ones who are treated. There's no question about that. I mean, you and Janie showed that. Right. But I'm wondering about the ones that don't ever have to have treatment.
I think so, yeah.
The men may be a good model system for this.
Right. Mhm. And along those lines, we have several CML patients who are BRCA1 mutation carriers who, again, they're double cancers. They get the CML first, and then they've gotten breast cancer.
After they were treated for the CML? Yeah.
So I have a question.
Uh-huh.
I'm not a hematologist.
Uh-huh.
Most of these syndromes are recessive, right? At the cell level, they're recessive. At the inheritance level, they're
It's
dominant.
very interesting.
Except for SAMD9N and SAMD9L, which are dominant in all stages. And in there, you have the incredible reversion
Yes.
of mutations, either
Yes.
with a secondary mutation in
Yes.
CIS or UPD.
Yes.
Do you see that sort of thing with other, I don't know, do you see that? with
We
these
do
other
not see
syndromes?
that phenomenon with other syndromes, but I think your observation is very relevant. We call these syndromes autosomal dominant because of the inheritance pattern, and we say that autosomal recessive conditions like Fanconi anemia, they are recessive, meaning the heterozygous state does not confer risk. But we see heterozygous Fanconi mutations all the time and other chromosome breakage syndromes that are supposed to be recessive all the time. And the Australians also see this. And so, Colin and I were talking about this, that it's just harder to show because the risk is smaller. So you need a much larger sample size in order to show because the effect size is small. And I do think moderate risk alleles matter.
Oh, yeah.
And so, it's going to take large studies to show that heterozygous Fanconi mutation carriers have a cancer risk. I also think, as we were talking this morning, I also think that mutations can have different effects in different tissues. And so what can be a tolerated variant in one tissue could be deleterious in another. So it's complicated. We know people are somatic mosaics. And
Certainly are for Sicilian and
Right.
which brings up another possibility. If you have siblings with that disease, you don't necessarily not use the person who also inherited the germline
Yeah.
mutation, but developed a somatic reversion.
Exactly. I mean, we could spend a whole hour just talking about SAMD9 and D9L, and I tried to give a more general kind of talk, but they're absolutely fascinating syndromes. Yeah. You had a question in the back? Yeah.
Yeah, thank you for coming to visit. That's a great talk. I think I heard you talk about P53 knockout mice that you put on the shelf and through phlebotomy.
BRCA1, that mice that have been knocked out in the bone marrow for BRCA1.
And through phlebotomy, you either generate mutations associated with malignancy or generated malignancy itself?
So if you knock out BRCA1 in the bone marrow and just leave the mouse on the shelf, the steady state hematopoiesis it's not it doesn't drive any kind of heme malignancy generation. I mean, it might if we had huge numbers of mice, but keeping the colony at a reasonable size, you don't see it. But as soon as you start bleeding those mice, which does drive a stress hematopoiesis and drives the hematopoietic stem cell into replication, I think what's happening is because BRCA1 is not present, backup DNA repair pathways are present. Those are error-prone pathways, and then errors get introduced. That's what we're studying now.
So is it just simple increases in hematopoiesis, or are you inducing anemia? Hematopoiesis.
So we don't induce, it's complicated. Well, let's see. No, you're correct. It's complicated. We're inducing enough of an anemia, yes. And so it's a complicated situation. And then what's the chicken, what's the egg, what's actually driving it? It's complicated, yeah.
Just related to Brad's question, what if you just give the mice EPO or TPO or something?
We haven't done it. We haven't done it yet, yeah. It's a great question. It's a great experiment to do, yeah. Eli?
It's a very naive question. Can you there are these donor-derived relapses after transplant? Yes. It was XX and now it's X.
Yes, that's right. We've seen donor-derived leukemias in two contexts. Germline mutations where the germline mutation was not recognized in the donor, or clonal hematopoiesis in the donor. And in that case, it's kind of similar to the mice. Generally, what you're doing when you transplant is taking a small number of stem cells from the donor and asking those cells to replicate into an adult body. So it's a huge replicative stress on those cells. So far, all of those types of donor-derived leukemias that are in the recipients, the donors are fine. It seems like the one-time GCSF stimulation of the marrow is not enough of a drive to induce leukemia, but asking that small number of stem cells to recapitulate a bone marrow is a huge replicative stress and can lead to donor-derived leukemia. So those are the two scenarios where we've seen donor-derived leukemia.
And I think, Lucy, the associations with donor-derived leukemia, the observation is a higher incidence if the donor's older. And so, there is a moving sort of target.
I know, and I feel bad because I wrote an opinion piece one time saying that the CIBMTR was ageist and should raise the age of their donors. And now that I see this, I feel like actually maybe they should lower it, or let's find out who has clonal hematopoiesis and pick somebody who doesn't. But of course, Colin would say everybody has it. If you look deeply and hard, you'll see it in everybody.
The argument against that is there's certainly a lower hanging fruit, which is the germline people, right? I mean, we're not...
Exactly. And the other thing is, when we've seen these donor-derived leukemias, they happen many years after the transplant. And if you hadn't transplanted that person, they probably would have died of the leukemia. But of course, then somebody would say, well, you could have done a cord, and they would have, So I just think I don't know how many people are here are from the transplant community, but generally the transplant community doesn't want to hear about germline mutations. They see us as a thorn in their sides because we're going to delay the transplant. You're all laughing. I don't know. Huh?
The mutation won't go away because you don't know about it.
Well, that's what I try to say, but they just want to, not think about it.
10 minutes over, so bring the conversation up to the front.
Okay, thank you. Sorry I went over.